The remarkable similarities between a recently leaked 2018 proposal from EcoHealth Alliance head Peter Daszak and the defining characteristics of COVID-19 raise new questions regarding the origins of the pandemic. Daszak's proposal detailed how he and collaborators from the Wuhan Institute of Virology plan to create entirely new coronaviruses through the synthetic combination of pre-existing virus backbones, creating entirely new viruses which would have no clear ancestor in nature. Lack of ancestral lineage is a notable feature of COVID-19. The proposal also detailed plans to genetically engineer a human-specific furin cleavage site, the defining feature of COVID-19, into coronaviruses. Daszak further proposed to create a novel nanoparticle coronavirus vaccine. At the same time, the Wuhan Institute of Virology had developed a remarkably similar vaccine utilizing what appears to be the same technology. Hi everyone, and welcome to Truth Over News with Jeff Carlson and Hans Manka.
In the early days of the COVID-19 pandemic, Anthony Fauci took part in a teleconference where participants were told by scientists that it appeared highly likely that the virus originated from a lab. Within minutes of the teleconference's start, one of the participants emailed a question to the group. We need to talk about the backbone too, not just the insert. While it was known that Fauci's teleconference group was focused on the insertion of a furin cleavage site, the reference to the virus's backbone had been something of an enigma. Up to now, the group's interest in the virus's backbone had not been fully understood. Daszak's proposal now provides a strong indication that Fauci's teleconference group suspected that the backbone of the virus itself was engineered. The proposal, dated March 27, 2018, sets out Daszak's plan to create entirely new coronaviruses through the synthetic combination of pre-existing viruses. It describes how those viruses were going to be made more virulent in humans by the insertion of a furin cleavage site, a feature that distinguishes COVID-19 from all other SARS-related coronaviruses. Daszak's proposal, which was leaked by a whistleblower to Daszak, an online community of researchers investigating the origins of the pandemic, has since been authenticated by Linfa Wang, one of the scientists involved in Daszak's proposal. The new revelations contained in Daszak's proposal have far-reaching implication for the ongoing search of the virus's origins, and they also help to decipher what was discussed at Fauci's secret teleconference, where the natural origins narrative was established.
There have been a number of puzzling features surrounding COVID-19 that set it apart from any other known coronavirus. COVID-19, which was immediately well adapted to transmission in humans, is surprisingly poorly adapted for infection in bats, indicating significant evolutionary hurdles in acquiring its human transmission feature through natural evolution in bat populations. This adaptation for human transmission was highlighted in a recent study in the science journal Nature, which noted that the COVID-19 virus was uniquely adapted to infect humans, as it exhibited the highest binding to human ACE2 of all the species tested. COVID-19 is also the only known SARS-related coronavirus that contains a furin cleavage site, the defining feature of COVID-19 that sets it apart from any related viruses. The furin cleavage site is also the key to the virus's pathogenicity in humans. This feature of COVID-19 was often puzzled over by scientists early in the pandemic. While it has been argued that a furin cleavage site may have sprung up naturally, there is no evidence of this ever happening. However, furin cleavage sites have been inserted in a laboratory setting. A furin cleavage site can be inserted using seamless technology, a process that would leave no evidence of the lab insertion. Jingli, the director of the Wuhan Institute and one of Daszak's proposal partners, supervised a thesis in April 2017 that detailed how her lab was already performing experiments that replaced the S gene without traces on bat coronaviruses.
Both Daszak and Jingli have gone to great lengths to avoid discussion of the furin cleavage site. Daszak has publicly downplayed the presence of the furin cleavage site, while simultaneously failing to admit that he proposed to insert such sites into coronaviruses in conjunction with the Wuhan Institute of Virology. Jingli described the features of COVID-19 in minute detail in her highly influential February 3, 2020 article in Nature magazine. However, she conspicuously left out any mention of the virus's furin cleavage site. She's omission is striking, as the presence of the furin cleavage site was immediately visible from looking at COVID-19's genomic sequence, which was released in January of 2020. The significance of the site was acknowledged by Vincent Racaniello, a virologist and natural origins proponent who frequently features Daszak and Daszak's collaborators on his weekly podcast. Only days after Jingli had concealed the existence of the furin cleavage site, Racaniello conceded that the insertion of a furin cleavage site into another virus, the H5N1 influenza virus, showed that it made the virus more virulent. In what looks like a Freudian slip, he also wondered if the insertion of a furin cleavage site allowed a bat coronavirus to gain the ability to infect humans.
Despite the presence of a unique furin cleavage site, COVID-19 generally resembles other bat coronaviruses. But conspicuously, the virus's genomic backbone does not actually match any previously recorded backbone. This lack of a traceable backbone has been a primary argument used by natural origins proponents in their claims that the virus could not have been engineered. Christian Andersen, a Fauci-funded scientist who has become known for promoting the natural origins narrative, specifically cited this in his proximal origin paper when he stated that the genetic data irrefutably show that SARS-CoV-2 is not derived from any previously used virus backbone. But there are several problems with this argument. The lack of a previously used backbone as the basis for genetic engineering does not preclude the use of an unknown one. The natural origins argument presupposes that all scientists diligently post their genomic backbone sequences to public databases.